So firstly, I'd just like to welcome everyone today to our first webinar in our series of 4 condition-specific webinars. That is an extension of our Retina 2020 virtual event. So thanks everyone for joining today. Just before I hand over to our CEO, Fighting Blindness' CEO, Kevin Whelan, to give the official welcome, I would just like to go through a couple of housekeeping notes before we get kicked off. So as you'll notice that this webinar is hosted over our Zoom webinar function. So that means that you can't turn your camera on or unmute your microphone over the course of the webinar. So we did ask people to submit their questions in advance if possible, and we have them on file, and thank you very much for everyone who did so. But of course, if there is questions that come up for each of the speakers, you're more than welcome to put it into the Q&A box, and we will get to as many questions as we can within the allocated time. There's also, just to let you know, that this webinar will be recorded and it will be available in the coming weeks afterwards for anyone that mightn't have been able to make it or just to watch back. And we'll be in touch by email about this, the details of this, and also with a short feedback survey that we kindly ask you to fill out and get back to us to always improve. So also, just before I hand over to Kevin, I just like to thank our 3 sponsors for this session: ProQR Therapeutics, Novartis, and Roche. So thank you very much for supporting Fighting Blindness in this session. So Kevin, if I could just ask if you could switch on your camera and your mic and give the official welcome. Thank you.
Thanks very much, Niamh. It's a pleasure to welcome everyone this evening to our education webinar. And I think it's really good that we've been able to do this because it honors a commitment that we made at our Retina conference last year. Because traditionally at our Retina conferences, we would have breakout sessions that would be condition specific. But obviously with COVID, that wasn't a possibility this year. But our commitment was that we would follow up with these webinars. And it's my pleasure and honor to launch our first webinar. And obviously, Niamh has thanked our sponsors, Novartis, ProQR, and Roche. But I hope that it is also to the uppermost in our minds to thank our research team who have pulled this and compiled this webinar series. There's a lot of work has gone into it, and I'm very appreciative of what Niamh and Rachel have done in that regard. Also, I think it's very opportune to thank our founding fathers, the people and mothers, the people who actually started this organization and this charity, and they would always have hoped and aspired to treatments and cures. And I think it's a great day that we look at gene therapies being available. And during the presentation by Ben Shaberman, we'll hear of other treatments and therapies that are in the pipeline. So I think a big thank you to those who established Fighting Blindness, because this webinar and this information and the treatments and the advocacy and the patient-centered approach would not be there without that ethos having been established for day one in the organization. And I suppose for the staff that work in Fighting Blindness, for the board of directors and all the various advisory committees we have, it's great that we have that legacy and that we are honoured to be custodians of that legacy into the future. So we thank everyone in relation to that. Also, during the course of our webinar today, you'll hear from various speakers: Jackie Turner, Genetic Counsellor; Ben Shaberman, Senior Director of Scientific Outreach and Community Engagement with our great friends Foundation Fighting Blindness; Professor David Keegan, Clinical Ophthalmologist; and Alison, Reynolds, University College Dublin. We want to thank our contributors, and we are looking forward to addressing questions that have been raised in advance of the webinar, and we hope that you enjoy the information that you receive, and I hope that it provokes debate, discussion, and questions. And I thank you all sincerely on behalf of Fighting Blindness for participating in the webinar. And with that, I will hand back to Niamh, who will start the presentations with the overview of genetics and inheritance patterns as pre-recorded and delivered by Jackie Turner, Genetic Counsellor. Thank you, Niamh.
Great. Thanks, Kevin. Just bear with me now. I'm going to share screen. Yeah, so this, oops, sorry.
So I'm Jacqueline Turner. I'm a genetic counsellor in the newly formed Clinical Genetic Centre for Ophthalmology in the Mater Hospital. The unit was developed in response to a need for a genetic counselling service to return genetic test results to patients and their families who were tested by the Target 5000 team at the Mater. The Target 5000 study began in response to a new emerging approach in medicine called precision medicine. Precision medicine allows treatments to be selected based on the genetic factors underlying a patient's disease. Gene therapies, where you are putting a working copy of a gene back into the eyes of patients that are missing that gene, is an example of precision medicine. All patients participating in the Target 5000 study have samples taken for genetic analysis. This is performed first in the Trinity Research Laboratory, and there they find the spelling mistake, which they believe is causing your eye disease. Once we have that, we can then send your sample over to our diagnostic laboratory where they can confirm the result. The diagnostic lab will also produce a genetic report. We then meet as a team and to determine if the patient's photos and eye examinations and family history fit with what has been found in the genetics report. We also check that the changes found are classed as disease causing. This is very important as we have millions of changes in our genetic material, most of which are harmless, some are beneficial, and only some are disease causing. And we need to be confident that the changes found are causing your condition. Genetic counselling involves helping patients understand the cause of their eye condition and how it is causing their visual impairment. I'll also discuss how the eye disease is inherited and what are the chances of a patient's children developing the condition, and who else in the family could be at risk of developing the condition. Once we are confident that the changes found is disease causing, we can offer testing to those who are at risk, if they want to know, to determine if they are likely to develop the condition in the future. We are at a very exciting time in medicine with therapies that are emerging that can halt the progress and even improve the vision of those with genetic eye disease. And we hope in the future that these therapies may work at curing eye disease. We all have genes which make proteins which direct our body's processes and ensure that our body is working and functioning normally. The new genetic testing allows us to examine many genes at the same time. There are over 300 genes which we need for our retina to work and function normally. With the new genetic testing methods, we can screen all these genes together to see if we can find a mistake in one of the genes which is causing our visual problems. We can then check that that mistake is actually disease causing. Often the changes found in the genes are unique to the family or haven't been described yet. We are very new to this. The human genome was only first sequenced in 2003. By finding the gene involved, we can learn more about how a particular gene is causing the condition. For example, you may have been diagnosed with retinitis pigmentosa, but there are over 80 genes which can cause retinitis pigmentosa. And perhaps some genes cause a late onset type. Other genes might cause an early onset type. Some genes might cause a slowly progressive type. And for other genes, the progression might be quite quick. So in order to know how a particular gene is causing your condition, it would help us in the future with your prognosis. Knowing the gene helps us to determine how the condition is also inherited. Everyone who develops a visual impairment throughout their life will grieve their missing vision in some way. It is quite normal to feel sad that you do not see as well as you used to. The important point here is that you're not alone. There is a team of people available to support you and to try and enable you to accept your loss and to move on with your life, concentrating more on what you can do rather than what you can't, so that you can live your life to the full. It is important to remember to do what you enjoy, and if you really can't do that, to investigate other interests and hobbies that you might enjoy. There is also much more support available than there was in the past in education and in the workplace. And technology has enabled those with visual impairments to participate as fully as possible in what is going on around them. There are so many gadgets and apps available to assist you. There is always a solution. If you are part of the Target 5000 study and you wish to be seen for genetic counselling, please ask your clinician to refer you to me. Thank you.
Thanks, Rachel, for saving the day there. So there might be a couple of questions that come in for Jackie after that presentation, but this is a pre-recorded talk, so maybe these questions can be answered by David Keegan a little bit later in the session. And just to move on to our first live speaker of this evening, I'm very happy to introduce Ben Shaberman. As Kevin had said, he's the Senior Director of Scientific Outreach at Foundation Fighting Blindness. And a lot of you might remember Ben from he gave a talk at our Public Engagement Day in November, and we're absolutely delighted to have him back to give an overview of the research and clinical trials updates. So Ben, I think you're set up to go, but thanks very much and we welcome you.
Well, thank you, Niamh, for this opportunity. Can you hear and see me okay?
Yeah, we can.
Okay, great. Well, again, thanks, Kevin, Niamh, the whole Fighting Blindness Ireland team for inviting me. It's always a pleasure and a privilege to partner with you and present the research. I only wish that I was live in Dublin instead of here on the East Coast in the US, but maybe in the next year or two I can visit. I'm going to cover a number of research projects that are in the clinic or close to the clinic for RP, LCA, Usher syndrome, and choroideremia. Some of these projects are cross-cutting, so they work independent of the gene causing the disease. But what I wanted to emphasize is I can't cover everything that's in a clinical trial right now just because of time, but that's a nice problem to have. There are about 40 clinical trials, more than 40 underway in our space. And many of these have come to fruition over the last 10 to 15 years. So it's a very exciting time in our space. And again, it's a privilege and an honor to be the messenger to tell you about all the great things going on. So let's get started. I wanted to first talk about gene therapy for X-linked RP. As you heard in the video before me, with gene replacement therapy, you're essentially replacing the disease-causing gene, the mutated gene, with a healthy gene. And one fairly common cause of retinitis pigmentosa is the X-linked form. And most people with X-linked RP have mutations in a gene called RPGR. And excitingly, there are 3 clinical trials that are now underway to replace the RPGR gene, gene therapies for X-linked RP. These trials are either in phase 3 or they're moving toward phase 3. And the companies that are conducting these trials include Applied Genetic Technologies Corporation, AGTC, Biogen, and MiraGTx with their partner Janssen. Now, in earlier stages of these trials, they all reported some vision improvements in retinal sensitivity, in some cases visual acuity. So we're very encouraged by the progress for all these companies. And I think we're very confident that at least one of these will make it through phase 3 to regulatory approval. Now, there is a fourth company that is conducting a gene therapy trial for X-linked RP. They are called 4D Molecular Therapeutics. They're at an earlier stage. They're in phase 1/2. What makes them a little different is they inject the gene therapy into the middle of the eye, into the vitreous. These other companies that I was talking about, their gene therapies are administered underneath the retina through subretinal injections. So long story short, the intravitreal injection is a less invasive, safer approach. And so we're excited about this work from 4D because injecting a gene therapy underneath the retina is not a trivial matter. It is a surgical procedure. So if we can do it through an intravitreal injection, which can be done in an outpatient setting, that would be a big benefit. So that's X-linked RP. Another condition for which progress has been very encouraging is choroideremia. And Biogen, which acquired a company in the UK not too long ago called Nightstar, has a gene therapy clinical trial for choroideremia in a phase 3 study. It has sites in both Europe and the US. They have completed enrollment in their phase 3. About 170 participants have been enrolled in 18 sites. At an earlier stage of this trial, visual acuity was maintained or improved for most of the patients, for over 90% of the patients. So that's an encouraging result. And now we're just waiting to hear what the phase 3 results are now that they've completed enrollment. Hopefully, if those are good, they will seek regulatory approval. And again, the company that I mentioned, 4DMT, who's developing an X-linked RP gene therapy through that's administered through an intravitreal injection, they too have a choroideremia gene therapy trial that is in a phase 1/2 stage. That's only in the US at this point. They have yet to report results since they're still at an early stage. So good things happening for choroideremia. Now, I wanted to talk about a company, one of our partners, who has a number of gene therapies in preclinical development. These aren't in clinical trials just yet, but they're addressing a number of challenging conditions. These include RP caused by a common rhodopsin mutation, Best disease, LCA caused by a common CEP290 mutation, Stargardt disease, which is caused by mutations in ABCA4, and also a common mutation in USH2A. Now, what makes many of these genes challenging is that they are too big to fit in the viral containers that are used to deliver them into retinal cells. So this is true for CEP290, ABCA4, and USH2A. And on the right side of this diagram, of this slide, I should say, you see this knobby, colorful ball. This is a virus, a viral container, and this is the delivery system we use to get copies of these genes into the retina. But these containers have a limited capacity. And for the 3 genes I just mentioned, they're too big to fit in these containers. So what Iveric is doing is they're partnering with actually UMass to shrink these genes down so they fit in the containers, but yet they're functional. They make proteins that are functional. So we're excited about this work because it's offering gene therapy to people who have these more challenging genes. Now, the rhodopsin gene at the top that I mentioned that's at the top of this slide, what makes that gene challenging is that it, when it's mutated, it makes a toxic protein. And in addition to replacing it, you have to knock down that toxic protein so that that protein, that bad protein isn't made. It's a two-step process. So that makes the rhodopsin, specifically the P23H mutation, a little more difficult to address. But again, Iveric Bio is teaming with University of Florida to move that into a clinical trial, hopefully next year. So a lot of great work from Iveric Bio. Now, moving on to a relatively new gene therapy trial for a form of LCA. This particular form is caused by mutations in a gene called GUCY2D. And this came out of the lab of Shannon Boy at the University of Florida. She co-founded a company earlier this year called Etsena, and they launched a clinical trial for a gene therapy for this gene. Now, kids who are born with this condition, LCA, again, GUCY2D mutations, they have very severe vision loss. However, their retinas do retain cellular structure. They do have some surviving photoreceptors. So the idea is to rescue and restore these surviving photoreceptors. So recently, Shannon and Etsena reported some encouraging early results for people with advanced vision loss, their first 3 patients. They did have some modest vision improvements at the lowest dose. And so that's encouraging as they move into higher doses for younger patients with better remaining retinal structure. Now, Shannon is also developing a gene therapy for Usher syndrome type 1B. That gene, it's called myo7A, is too big for those viral containers I was talking about. So she's developing a 2-vector system to deliver that large myo7A gene. So it's delivered in 2 containers, and when it gets to the retinal cell, when it gets to the photoreceptor, those 2 pieces are combined to make a complete gene. That is not yet in the clinic, but she's had good progress in developing this, and hopefully that will be in the clinic soon. And this approach, the dual vector approach, like the mini gene approach, can potentially apply to multiple retinal conditions where you have those large genes. Now, I want to switch gears a little bit, and instead of talking about gene replacement, I want to talk about RNA therapies. So a quick primer on RNAs. So your cells have a genetic messaging system, and it's actually these genetic messages that your cells read so they know how to make the proteins you need. And these messages are called RNA. And so the mutations that are in our DNA are also carried in our RNA. And this company ProCure out of the Netherlands, and one of your conference sponsors, has developed an approach to mask certain mutations in RNA. And these masks, this is a very long term, are called antisense oligonucleotides. And essentially, they're tiny pieces of genetic material that cover the mutation in RNA, so your cell can read that RNA and make a functional protein. So excitingly, they have had excellent results for this approach in a phase 1/2 for a CEP290 mutation, a pretty common mutation that causes LCA. So they moved this approach into a phase 2/3. They've also had good results for the exon 13 region in the gene USH2A. That mutations in that region can cause Usher syndrome and even non-syndromic RP. So thanks to good results in phase 1/2, they are moving it into a phase 2/3, hopefully by the end of the year. And then finally, they are also addressing the P23H mutation in rhodopsin, which causes RP. That's still in a phase 1/2. They haven't reported results just yet. And if you look at the bottom right part of this slide, you can see an image. That vertical red line is actually a depiction of the treatment masking the mutation in a piece of RNA. So moving on, I want to introduce yet another approach to addressing a genetic mutation. And this approach is called CRISPR-Cas9, and it's basically gene editing. So again, instead of replacing the gene, what this approach does is it goes in and, like a pair of molecular scissors, it cuts out the mutation. So instead of delivering a whole new gene, you're just cutting out the bad part in the patient's existing gene. So several months ago, a company called Editas launched a clinical trial for a CRISPR-Cas9 therapy for this common CEP290 mutation that I've been talking about. That's in a phase 1/2. This happens to be the first CRISPR-Cas9 treatment for any condition that's been applied to the human body. So we're excited that they chose retina first. The retina is a good target for many reasons. It's very accessible. So we're still waiting to hear results, but we're encouraged so far. Editas also is in preclinical development for USH2A exon 13, and again, that rhodopsin P23H mutation that causes RP. So all of the genetic approaches that I've talked about so far are addressing specific genes or specific mutations. What I'm about to talk about is gene agnostic. It's designed to work regardless of what gene mutation is causing your disease. And this is being developed by a company called Sparing Vision in France, along with University of Pittsburgh Medical Center. And this therapy expresses a protein that keeps your cones healthy. Again, it's gene agnostic. Now, this protein is called rod-derived cone viability factor. It's actually similar or just like a protein that rods normally excrete that helps keep your cones healthy. Well, that's what this gene therapy does. And the idea is if you can preserve cones in people, you can help them maintain the vision that's most important to their activities of daily living. Cone photoreceptors give us visual acuity, the ability to read and recognize faces. They give us vision in lighted settings and color vision and central vision. So saving cones would be a wonderful thing. This trial is expected to launch late this year. It would move into the trial for RP, and it can potentially help people with RP, Usher syndrome, and again, situations where you have surviving cones that you want to save. Okay, we're going to talk about yet another gene therapy approach, and this is different, again, from everything we've talked about. This approach is called optogenetics. But before I talk about optogenetics, I want to talk about the patients for whom this approach would be most applicable. So on the bottom left part of this slide, I have a side view of the retina. And toward the top of this side view are the long cells, which are the rods and the cones. And those are the cells that make vision possible. And I have this ominous X going through the photoreceptors, going through those rods and cones. And that's to signify somebody who has lost all their photoreceptors to an advanced retinal disease like RP or even AMD. So if you lose all your photoreceptors, by definition, you're blind. And all those genetic therapies that I talked about up till this point won't apply because there are no photoreceptors left to save. But some very clever researchers identified other cells in the retina that survive after photoreceptors are lost. And they are at the bottom of this image, and I have them circled. These are called ganglion cells. What these clever researchers did is they deliver a gene that expresses a light-sensitive protein to make these ganglion cells operate like a backup system for lost photoreceptors. So there are a few companies in clinical trials now for this approach: GenSight, Allergan, and also a company called Bionic Sight. They have an early stage clinical trial in New York State, in actually New York City, Long Island, here on the East Coast. And in their phase 1/2 clinical trial, they reported some encouraging results for the first 4 people that have received this treatment. These people can see some light, shapes, motion. One gentleman who was completely blind was washing the back of his wife, and he could see his hand against her back. Now, these folks are getting the low doses of the treatment. We're hoping that as they move into higher doses, these patients will have even better vision. The Bionic Sight approach is combined with these glasses that help enhance the visual information that's coming into these treated cells, these ganglion cells in the retina. So stay tuned for more on Bionic Sight and these other trials. So we're rounding the bend here toward the end of my presentation. I wanted to talk about a couple of stem cell therapies that are in clinical trials. These both are designed to work independent of the mutated gene, and both involve stem cells that have almost fully developed into photoreceptors. Stem cells are a clean slate. You can make just about any retinal cell you need, or any cell type in the body, out of stem cells. We're most interested in making photoreceptors for obvious reasons. So these are precursors to photoreceptors. So in the JSight trial, which launched in Southern California and now is in multiple sites throughout the US, their cells are injected into the middle of the eye, and the hope is that these cells can preserve vision, maybe rescue a little bit of vision, but preserve vision. We call this approach neuroprotection. The idea is to save and perhaps rescue cells that are still alive in the retina. So in the phase 2 trial, JSight reported some encouraging results, especially for a population of patients that entered the trial with better vision. They had about 3 lines of vision improvement after receiving this approach, that subpopulation that, again, entered the trial with better eyesight. So JSight is receiving funding from the pharmaceutical company Santen, and they are moving into a phase 3 for Usher syndrome and RP. Now, another company that is using this same cell type as a treatment is called Reynolds. And Reynolds is a company out of the UK, but instead of injecting these cells into the middle of the eye, these cells are being injected underneath the retina, subretinally. And the idea, or the hope, is these cells will integrate with the patient's existing retina to replace photoreceptors that have been lost. They too are targeting people with RP and Usher syndrome. They are in a phase 2 study right now. They're expanding the phase 2. And thus far, most of the patients in the phase 2 have had about 3 lines of vision improvement, and that's been sustained out to 18 months. So Reynolds currently has sites in the US, the UK, and Spain. And hopefully, as this phase 2 trial is expanded and progresses, they will continue to get good results. and can move into a phase 3. Like J-Site, they are trying to identify the patients in the trial that are responding best so they can enroll those types of patients in their phase 3. So that concludes my talk. Again, there's a lot of other great work going on that I wasn't able to cover, both in clinical trials and moving toward clinical trials. If you want to learn more about these therapies, you can go to our website, fightingblindness.org. Just about all the trials underway in the US and most in Europe are on a website called clinicaltrials.gov. And if you want to get on the radar screen of companies and researchers conducting clinical trials, go to myretinatracker.org and enter your information, and you can potentially get contacted about clinical trials for which you may qualify. So that is the end. Thank you.
Thank you very much, Ben. That was great. And I know it must be very hard to cover such a wide range in a short space. So we appreciate that. There was a couple of questions that came in and some of the pre-submitted ones. So a question that we got quite a bit, I think you kind of covered it a bit, but for people that have an unidentified gene, and what is the kind of future advice for them in relation to clinical trials?
Sure, that's a good question. And we, that is the Foundation Fighting Blindness in the US, is very focused on developing gene agnostic therapies to help people, whether they don't know their gene or whether they know their gene, but there isn't a gene therapy or a genetic treatment being developed for it. As was mentioned before my talk, there are more than 300 genes that we know of that, when mutated, can cause a retinal disease. We're just not going to have genetic specific therapies for everybody. So the sparing vision, that neuroprotective therapy that I mentioned, that's gene agnostic. The optogenetic therapy, where you're restoring vision to people who have lost all their vision, that's gene agnostic. And the cell therapies are gene agnostic. One trial I didn't mention for RP and Usher syndrome is for an antioxidant developed by a company called Nacuity that we helped fund. And that trial is underway in Australia. It's an oral medication. That too is gene agnostic. So lots of great stuff going on for people regardless of their mutated gene.
Thanks a lot, Ben. And then there was just one other question that came in. Some of the results and therapies for Usher's type 2, can that ever apply to Usher's type 1, or is it completely separate?
So all the gene independent approaches that I mentioned, those can apply to whatever form of Usher syndrome, 1, 2, or even 3. Now, if you're getting into gene specific approaches, USH1 and USH2 are two different things. Usually there are multiple forms of Usher's syndrome type 1. There's 1B, I believe there's a C, a D, I know there's an F. Most people have USH1B. I mentioned that Shannon Boy is addressing that. I know 1C is being addressed. And USH2A, we have some emerging therapies that I talked about. So they're both gene specific or mutation specific and gene agnostic, but they are genetically different, each type of Usher syndrome. There are about 12 genes, each of which can cause Usher syndrome.
Okay, thank you very much, Ben. I think that's all we have time for today, and we really appreciate you joining us, and hopefully we can see you in person again sooner rather than later. Okay, thanks, Ben.
My pleasure. Thank you.
Yes, welcome. So we're going to move along to our second live speaker, who is another familiar face. Many of you will have heard Professor David Keegan speak at our virtual public engagement day in November. So we're delighted to have David join us again today. He's going to give us a clinical update in relation to these conditions. We did have a couple of questions come in regarding diet and how that relates to eye health for these conditions, as well as a couple of COVID-19 related questions. So I think David is going to address these and also some of the other questions. And if there is questions that come up, please pop them into the Q&A. So David, welcome and thanks very much for joining us today.
I think David might be on mute, is he?
That's my second fine of the day. Apologies. Thank you very much, Niamh and the Fighting Blindness team for inviting me to speak again. And I hope to all the attendees you don't get too tired. We are training up the next generation, so you won't have to put up with me for much longer. I would say I always enjoy Ben's talks when he gives them, and it really is a great overview and update on the landscape around clinical trials and therapies for retinal degenerations. I'm hoping that this talk now segues nicely on from Ben's, in that it will it'll show that what we're doing in Ireland to bring the Irish population with IRDs up to speed or into the mix to access these therapies. And really, none of that would be possible without the input of Fighting Blindness and all that they've done over the last few years, and particularly supporting Target 5000, which is a great collaborative initiative between the three clinical sites, the Mater, my own site, the Eye and Ear Hospital, and Belfast Trust. But in addition to Trinity, who's been at the heart of a lot of great discoveries around rare eye disease and genetic eye disease since the 80s and early 90s with Pete Humphreys, Jane Farrar, and the many people that worked there. So it's with great privilege that I get to speak about the clinical arm of what happens here now, because it's all well and good, all these things happening in the lab. It's got to transfer at some stage to realize that vision and dream of the founder members of Fighting Blindness that Kevin alluded to at the beginning, to realize a cure and a treatment and all the other things. So really, inherited retinal degeneration in a new era, and Ben's gone through this: gene therapy, gene editing, stem cells, neurotrophic factors, optogenetics, artificial retinas, which are a little bit out of the news, but then also sensory substitution. And apologies to those of you, I use these slides more as a prompt for myself, but on this slide, the sensory substitution, a tongue stimulator, which we haven't used greatly, but it just illustrates that there are many innovative ways to look at restoring vision, or that sense of vision. But we're going to concentrate a lot more on gene therapy in this particular talk. But when we were developing the clinical arm of Target 5000, and I have a lot to be grateful to Kirk Stevenson, a Target 5000 fellow who worked with me a couple of years ago. And really we looked to the UK and Graham Black's group as a model of what patients actually want, going back into that and liaising with Fighting Blindness and the family and Fighting Blindness about what so they want a clinical diagnosis, prognosis, and inheritance pattern. Practical support, signposting of services, education, low vision, and proper supports there, and psychological support to help adjust with diagnosis and loss. So really, these actually all cut to the pillars of Fighting Blindness: to cure, support, and empower. So it's very much along those lines the Target 5000 now is. So I've given up my old pathway map, and I've adopted Kirk's pathway map, and I've accepted that this is a better map. And apologies to those who can't see it, but really, it's that multi-phase, multi-stranded process that we have developed over the last 8 years or so for Target 5000 that moves patients through a referral process, a clinical assessment and phenotyping. They're put up on an electronic register, the Distiller register. We're looking to move off that into an open app register, which is interfaceable with the European reference networks. I'll talk a little bit about that. We do research grade genetic testing. If we get a positive result, we go and get clinically accredited genetic testing, previously at Manchester, currently at Blueprint in Finland, but now we're really looking to repatriate that to the soon to be accredited genomics laboratory at the Mater. So we'll repatriate all that genetic testing. And now with the help of Fighting Blindness and more recently the HSC, we've got our clinical genetics team. You've heard from Jackie Turner, the genetic counsellor. We also have Dr. James O'Byrne, the clinical geneticist who works with all of us. Jane Farrar's team, the clinical teams in the other hospitals, myself and the broader team, Julia Sue, who's the current Fighting Blindness Fellow. And we really, through that, then the patients can access genetic counselling. And Jackie talked really eloquently about that earlier on, about how important that is in the journey and the pathway to have the gene explained to you, the inheritance pattern, what it means to your family, what it means to you, and what it means possibly for a diagnosis and a possible treatment for your inherited retinal degeneration in the future. But we're also looked to bring in, and we're really proud that we've developed as part of this program, a bespoke care plan template, which is soon to be adopted by the European Reference Networks for Rare Eye Disease. That's something to be very proud of in this country. And Kirk has just recently got this paper published, and it'll come out in the next couple of weeks in Orphanet Journal for Rare Disease, this pathway model really to try and guide other small, medium-sized countries that may not have that infrastructure and service in place. We also introduce here the whole concept of comorbidity management, that there's other things, even if the disease itself is not modifiable, there's other things such as glaucoma management, cataract management, membrane management, macular edema that you can be doing when you're part of this system that'll optimize your vision ahead of going and reversing disease process. So this is just an example of a patient with USH1C. Ben alluded to that condition there earlier on. So we image the patients, phenotype them, we do detailed scans and that on these patients. We've also managed to develop adaptive optics here now in Dublin. This is in Brian Vonson's lab out in UCD in the Department of Physics. And Joe Carroll, a real friend of Fighting Blindness Ireland and a key researcher at FFB in the US, who Ben knows very well, has really helped us. So this allows single cell imaging and will be important when we're recruiting patients. And this is an image of the back of a normal eye with single cells. But this becomes important when we're mapping out patients who are suitable for gene therapy versus those patients who may need gene replacement treatment, such as stem cells that Ben talked about there. So this technology is now in Ireland. We've got all the tools available to us to start to target those patients who be suitable for therapy or suitable for clinical trials. I alluded to the Genomics Centre at the Mater. So this is funded by the Shaberman charity out of Monaghan and co-sponsored by the HSC and the Mater Ireland East Hospital Group. It has now already been accredited for cardiac genomic testing in 2020. And we've gone through the first 2 phases of accreditation for genetic eye disease, which they've passed. And we hope to commence testing for the Northern Irish, Julie Silvestri's group of patients in 2021. So again, this is all about repatriating that expertise onto the island so we can grow our clinical genetics expertise here, but we retain the support of those key collaborators in Blueprint and in Manchester, for example. So what happens after that? So you have patients who get the diagnosis, and this is from one of our other papers on a family with a condition called X-linked retinoschisis, which is a progressive degenerative condition. Well, we map out the gene and then we validate that gene when that happens by either in silico analysis and other testing. And this is where the clinical genetics service really starts to play its hand. And as Jackie said, that's a service available. It's open to all patients with inherited eye conditions, but specifically inherited retinal conditions. Get yourself referred into Jackie and James, and I'll happily see you myself as well in the clinic with Julia. So the initial funding is from Fighting Blindness and established in 2018. So we're going a few years now. One of the key things are the weekly multidisciplinary team meetings. So the clinical team and the genetics team with the administrative team meet to trash out the complexities of your genetic diagnoses. We have monthly Target 5000 meetings. These were suspended for a short time over COVID, but we hope to get these going again where we tackle those pedigrees. So patients who are under clinical care with Julie in Belfast or under Paul Kenneth's care in the Eye and Ear and our care in the Mater, but if they're all from one family, we can pull all that information together and resolve some of these diagnoses. And it's a key part of the team. And a number of cases have come through this pathway that we've upgraded from what we call a VU, or a variant of unknown significance. And the problem with a VU is we can't act on that clinically. So you may have a condition like Ben talked about earlier on, say a CEP290. But if that's a VU, we can't definitively say that condition is caused by that gene. So we've got to do a bit of investigative work in the background. And this is where Jackie and Jim with our colleagues abroad and in with Jane Farrar's team, they really work hard to say, can we get that upgraded? Is it really a VU? Is it really a likely pathogenic or a pathogenic variant? And if that happens, it's called a class 4 or a class 5, and that's your trigger point to treatment or talking about treatment. It's very important. So they upgrade the variants. We've also developed pathways for genotype phenotype mismatch where it looks like one thing, but the gene is something else. We've got to try and resolve that because there's a discomfort. We can't go putting you through gene therapy if they don't match up correctly. And we've also put a pathway in for managing secondary findings. So Julia has just published this in Eye Journal in the UK just last week. And it's a very important thing about what happens as we go to investigate your gene problem and we find another problem that's not related to the gene. Say like a BRCA mutation where it increases your risk of breast cancer or ovarian cancer. What do we do? So we've got a pathway in place to manage that. These are all the considerations when you put this service together. But also with the genetic service, we can start recommending therapies. And Jackie's become very skilled in guiding people on that journey as they come back to us into the clinic where we plan the next steps over the bespoke care plan. But what happens if we don't have a specific treatment? And we really are indebted to our partnership with the National Council for the Blind and the Health Service as we've rolled out this National Eye Clinic Liaison Officer Service, which we piloted alongside the Target 5000 program. And really, and this is Elaine Crossan, our adult ECLO, and we got Hilary Devlin in the Children's Hospitals, who's our pediatric eye clinic liaison officer, and they've transformed the service for patients because they understand the needs of the patient specifically, the vision needs, but also those support needs, school needs, college needs, employment needs, so that Elaine will help you navigate that whole system of supports that are available to you. And this has been really is a key addition to the whole team because we've got to manage patients holistically as well as targeted. We go back to that paper from Graham Black's group about what patients want and need out of this service. So part of it then is to link in, and this is again where you get the hybrid between the support and the research is the newly established NCBI lab service, which we collaborate with. And now we can get patients, and Ben talked about moving away from those gene-specific conditions when you have an gene-agnostic treatment. And a big part of that is technology. So with NCBI Labs, we hope to be able to bring in the latest innovations in technology to make them available for patients on the Target 5000 program. But again, ultimately, what most people on the call and on the Zoom this evening are really looking, how do they get access to therapies? So these are artificial retinas I have up there. This is stem cell treatment from the original Lancet paper, the AGTC trial with stem cells. This is from the gene therapy studies. And then if you talk about the X-linked retinitis pigmentosa, as Ben talked about earlier on, and we're very excited to be partnering in Ireland with Mira GTX at the matter about the phase 3 RPGE or gene replacement therapy for X-linked retinitis pigmentosa. So that's the overview, that's the structure, and that's what we spent the last 10 years spending all that money that people have fundraised for fighting blindness and bringing in. But it's important that you know it's achieving its primary goals. We have a structure and a pathway in place. We can never get to people quick enough. I get that when we see the questions come in, where's my gene result? What's happened to it? But because of restricted funding, we've had to go a very cost-effective route via the research lab at Trinity, and then going specific variant testing has kept the cost down. And Kirk estimates in his papers kept the overall cost for those who've gone through the program so far down by 46%. And that is significant. That's in the millions, that cost saving coming through. But if we just look at the matter, and apologies again for those with vision impairment on the call this evening, I like to use this inverted pyramid as a descriptor of the patients in care, because the definition of success is if we can convert that very pointed pyramid to a column. That means everybody who's in clinical care is on the electronic register, has had genetic testing, has had accredited testing, has received genetic counselling, has had family member testing, has had a care plan developed for them, and has been recommended for a clinical trial. But there's a couple of bits in this slide that I really want to highlight is down the end, is the explosion in accredited testing that we've been able to do. And that's been fundraised by the HSE and into the service. So now 327 of our 550 patients have had accredited testing. We've now delivered, and Jackie and her team delivered genetic counselling to 230 of those patients. That means we can release a care plan. So we're still in the final stages of fine-tuning that. And we're at the last iterative process with the European Reference Network. But to be able to give a care plan that's bespoke to the patient's needs, that will outline the eligibility for certain clinical trials for that particular condition. And that's a real goal of ours. And we've identified 40 patients with genetic alterations that are suitable to go into clinical trials like Ben talked about earlier on. Actually, I think that figure's actually gone out of date. We got some other genes back. Yes, I think that's gone up. And we've got 4 patients we've identified through this process with biallelic or PE65 disease. So that's the condition that is treatable by Luxturna, the only approved gene therapy treatment for any inherited retinal degeneration. That's important. I've got data back from Julie Silvestri in Northern Ireland. Unfortunately, I don't have the Ioneer data this evening, but Julie's represented this a little bit differently to me. So they have a total members affected, 686. They have about 560 up on the distiller as well. 503 up on distiller affected, 348 families. They've tested 245 of those, solved the mutation in 138 of those families, and they've completed counselling on 102 patients. So if you think that in the Belfast group and with ourselves, we've completed genetic counselling in over 330 patients. Now, I know that mightn't seem like an awful lot, given that we probably have about 2,600 patients, but given the haphazard way that patients could access genetic counselling in the past, we now have a firm structure in place. And now I've got quite a sizable business case put together for this genetic service so that we get additional genetic counsellors and geneticists to support patients with IRD in Ireland. But I want to talk a little bit about Luxturna. So here's the eligibility criteria for study 301, the key phase 3 study, and really is the eligibility criteria of the license for the drug. So more than 3 years old with genetic diagnosis of biallelic gene mutations. So we've got 3 patients over 3 and 1 patient under 3. Vision worse than 2/60 or constricted visual field. So we now know what we're looking for. And then they've got sufficient retina left that we can go and treat them. And we don't use this in clinical context. This is the mixed luminance mobility test, multi-luminance mobility test, and we're just going to use the thing full field stimulus threshold instead. Sorry for that complicated slide, but I put this up as a sign of how evolved and developed the whole program is, so we can present this to the funding agencies and the approval agencies, that we've got a full structure in place to select which patients are eligible for treatment and which are not eligible for treatment. And for those of you who can see the slide, down the bottom, there's a series of green boxes which essentially go for treatment in that context. So it's important that to get any of these things approved, they have to have confidence that we've got a system in place that is treating the right patients. We also need the infrastructure. It's a very busy slide, so apologies. We have the centres of expertise. The Mater and the Temple Street are orphanet centres of expertise, and via the European Reference Network Consortium application, which we're due to hear about later in the year, we'll collaborate with the Ioneer Hospital and Children's Hospital Crumlin. We've developed a genetic testing pathway. I've outlined that in those previous slides. And we also have in place the transitions of care from Temple Street to the Mater. So for children who are diagnosed under 16, that there's a formal transitions of care piece so that they don't get lost between 16 and adulthood. So again, other things that we had to put in place to make sure there's a gene therapy system. We've got a high-tech pharmacy expertise of the matter around oncology and biologics, and we were delighted that they joined us for this visit to the Dominic Fisher's group at Tübingen in December 2019. So we were able to develop a needs basis, a needs assessment on that to put the process in place. And I was on the phone to the chief pharmacist of the matter today to confirm all this is in place, and we'll be bringing in actually a new pharmacist to work alongside the excellent team there. We've commenced the onboarding training with Novartis, and this is all ahead of the approval, but this is how optimistic we are. The equipment is all in place for testing, to hold the drug, to give out the drug, and to deliver the drug. So that's all available by the full field stimulus threshold, which is due in the next couple of months. The surgery training, now the surgeons need to be trained multiply. So, multiple surgeons need to be trained. So it's not just enough to have David Keegan able to deliver this. So my colleagues, Paul Connell and Ian Dooley, are also there, and it's two consultants who'll be doing the surgery together. And I'm fortunate I'll have a colleague from the UK who's done these as well, will be coming in too. So we'll have the dry run testing of the pathway, and the new operating theatres available to actually go and do it. So we hope, if approved, to treat the first patients in Q3 or Q4 this year. So it's not just about the approved therapies. As Ben outlined, we've got a really exciting pipeline of therapeutics coming through. But once we've got that infrastructure in place, remember, Ireland has not delivered ocular gene therapy before. So we don't have that history like in Oxford or in Moorfields or Great Ormond Street. So we have to make sure we have all this in place. But now we're excited that we're linking in with Mira GTX and one of the leading companies in this area to be one of the sites for the phase 3 clinical trial. And we've already identified patients who can participate in this trial with X-linked RP. We're waiting for approval for Luxturna, I've told you that. And then the pipeline coming down the line then is a natural history study in RDH12 leading to a therapy, we hope. CNGB3, a chromatopsia phase 3 study, which we hope to start in 2022. And we're entering into discussions with ProQR for the treatment, the antisense oligonucleotide that Ben talked about earlier on, that we may be able to use this in the Usher's type 2 study for those patients specifically with an exon 13 alteration. But we're equally excited to be partnering with NCBI Labs about bringing in technological therapies or technological supports that will work for patients. And then in the future, we're hoping to bring in that optogenetic treatment. But all of these take a lot of setup, a lot of planning, and a lot of man hours. So we've self-funded, our clinical research nurses have been already 6 months working with Mira GTX to set up the ethics approval, the IRB approvals to get that in place to start that later in the year. But thank you for the talk. I'll bow out there. I probably went way over time, Niamh, and I apologize for that, but I hope that gave people a sense of where the structure, the process, and where Ireland is on that overall stage to start treatments and clinical trials.
Great. Thanks a million for that, David. That was great. I think we have a couple of minutes for some questions. I haven't checked the chat box, but there were some that you saw earlier. I don't know if you want me to pick a couple or
there's one here about, actually, I'll deal with this. Clinical trials of gene therapies for AMD. There actually are AMD trials coming in various guises. We will hope. I wasn't dealing with this specifically. Kathleen asked that question. I wasn't dealing with this specifically in this talk. But once we get our gene therapy pipeline up and running, we will be able to start those therapies. But as Ben said earlier on, subretinal injections are a far different beast to tackle than intravitreal injections. So once we have this system where we can manage to deliver subretinal gene therapy, I think we'll accelerate those other gene therapies coming into the country quite fast. Yeah. So when I bring up do you want to pick out another question there, Niamh, from the other ones that were asked?
Yeah, well, we had a couple of similar questions coming in about COVID in terms of people with RP, and a lot of questions about the vaccine. I don't know if you want to address that.
There's no evidence that there's a specific adverse effect at the moment from the vaccine.
Yeah.
However, this is a very new vaccine, it's a very new technology and coming out, and it's on the backdrop of rare conditions. So it's like a hematologist colleague of mine was asked the other day about patients with idiopathic thrombocytopenia, which is a rare platelet disorder, and the impact of the vaccine. Well, you've got a rare condition with a rare complication, so it's very hard to say. So when you say no direct evidence, so the advice we have really for everybody, and the ERNI advice, is actually patients with inherited retinal degeneration should have early access to COVID vaccine, because the nature of the condition, they're more at risk of contracting it because the distancing and contact is far more difficult to avoid when you've got a significant vision impairment.
Sure. Thanks very much, David. And then there was just a couple of questions about diet in terms of health of the retina for RP, and if you had any.
Yeah, broadly vitamin A, but avoid it in conditions like Stargardt's disease. So it'd be very specific. So talk to your doctor with your particular condition and what particular dietary advice you would have for that. So it's not a straightforward one nutrient or one vitamin is great for everybody. It's just to have a specific conversation. Can I address one question actually that did come up here, because it was asked by two people, Elizabeth and Robert, just about the genetic testing. So people were tested a couple of years ago on Target 5000. So you should have got from all the centres send out update letters about where they are on that pathway. So they're broadly 4 different types of letter. So one is you had your test sent to Trinity for research grade testing, but it has not been done yet because it's a slow and a particular. I'm not going to go into all the details, but it's a cheaper, it's a more cost-effective way for us to do it, but it's slow the way it gets done. The second letter is you've had your research grade testing, but it's come back as negative and further research is going to be done on that. The third letter is you've had your research grade testing, it's positive, it's now gone off for accredited testing. It has to go to an accredited laboratory before anybody can give you that result back. Or the fourth type of letter is you've had a positive test, it's gone to an accredited laboratory, we will be in touch with you for a genetic counsellor consultation for that to happen. So you should have one of those letters going out to where you are in the pipeline. But we are targeting direct accredited testing for specific groups where we think there might be an intervention that we can do more quickly. I hope that answers the question.
And just to echo in terms of what David has said there, if you haven't received any of those letters, we'd encourage you to make contact with us because we'd be very keen to try and help progress the issuing of the letter. And we would support you in following that up. So please come back to us if you need further help.
Yeah, okay, I would agree with that, Kevin. And you know, that system is there. Fighting Blindness have funded that. You've funded it through your fundraising. It's available. It's available for you. Please use it.
Great, thank you very much, David. I think that's all we have time for today, but thanks again for joining us, and that was very informative. So moving on now to our last speaker of the evening, last but not least, is Dr. Alison Reynolds from UCD. Alison is going to talk to us a bit about today Charles Bonnet syndrome, including an update on a recent survey that was conducted that she's going to explain. But just to mention on that note that Fighting Blindness has a VIP network, and that's for any visually impaired person that wants to be involved in research. And just for anyone that is a member of that, we're going to be in touch quite soon. But also for anyone that doesn't know about it or would be interested in finding more, please get in contact with us either by phone or by emailing research@fightingblindness.ie. Okay, Alison, if you want to share your screen whenever you're ready. Thanks a minute.
Thank you very much, Niamh. So hold on, let me just share here. Okay, so I won't take too much of your time, but I wanted to talk to you just about Charles Bonnet syndrome and a survey that we've developed for it in conjunction with Fighting Blindness. So I think some of you may know me. My name is Alison Reynolds and I'm a researcher. I run a research team looking at early onset severe retinal degeneration in University College Dublin. So I'm going to talk to you about this condition, and I kind of feel a little bit like I am maybe the least expert on this in the whole Zoom call. So feel free to share your kind of comments. That would be great. Okay, so a little bit just about this. Okay, so I don't know if many of you have heard of Charles Bonnet syndrome. So Charles Bonnet, I'm going to try and narrate the slides for you, was a philosopher and scientist who lived in the 1700s. And he actually described a condition which his grandfather had. So the condition the grandfather had, he was blind, but he could still see things. And what he could see was he could see things which weren't there. He could see people, he could see flowers, he could see carriages, so vehicles of the day. And this condition was written down over 260 years ago. It was described. And essentially what we now know is that Charles Bonnet syndrome causes a person whose vision has started to deteriorate to see things that aren't real. So we call these visual hallucinations. They're not associated with any mental condition, so nothing like that, but they can be simple patterns or they could be very detailed. So images of events, images of people or places. And they're only visual, so they should be just things that you might be able to see. They might be moving, they might be in color, they might be black and white, but they shouldn't involve any other sensations. So, not a sense of smell, not hearing. Now, Charles Bonnet syndrome seems to occur across all different types of visual impairment. So, you maybe have visual impairment from an injury. It may be an inherited condition. The thing in common seems to be that it's more, I suppose, more common in people who've lost more vision. And it's described in the literature as rare. Now, we think that it's described in the literature as rare because people are not very keen to come forward and tell people that they have visual hallucinations. And we think it's actually been really under-described. So why does it happen? So we think that it happens because as sight is lost in the brain, that we no longer get those nerve messages. So we no longer get messages going from the eye to the brain. And so our brains are quite clever. And so what they like to do is they kind of make up their own messages. So that message is gone and the brain kind of makes its own message. And so we think that it's kind of like the brain filling in gaps in sight loss. That is the gaps, I should say, that are there by sight loss. And one way you could maybe think about it is a phantom vision syndrome. So kind of similar to if you had lost a limb and you could still feel it there. It's kind of like phantom vision, where the vision is lost, the brain is kind of trying to fill that in. Now, there tends to be two different types. So number one is more simple visions that people see. So things like patterns or shapes. And the second thing are more complex. So things like people or animals, perhaps objects. So I'm just going to go on to the next slide here. And what I've done here is I've kind of drawn, not personally, I've kind of taken some images here of apparently what people can see with Charles Bonnet. So artists have kind of drawn what people can see with Charles Bonnet syndrome. And sorry, let me go back there. Apologies for that. And so on the left-hand side is a picture of a woman, kind of cartoon character sitting on a bus. And she is seeing kind of geometric shapes in front of her remaining vision. And she describes that sometimes she also sees people emerging from snowy verges, or an image that she'd seen many years ago. Now, on the right-hand side here, what I've shown is it's kind of a picture of a bathroom here. However, everything kind of looks reasonably normal, except you can see an image of some unusually dressed people who seem to be in the sink. And I suppose this is kind of maybe kind of what people with Charles Bonnet syndrome have described, where, it's a normal landscape, but they can see kind of unusual things in the landscape. And the third one that I have here at the bottom on the right-hand side is just a picture of a street scene, and then a picture of a street scene with phantom images across it. Now, in this case, the street scene is showing kind of images of animals across it. But I know that some of you have described to me how, maybe instead of an image, you might see additional vehicles, or you might see a wall where there's no wall. So the Charles Bonnet syndrome may affect any ability to navigate even with remaining vision. Okay, sorry, just trying to move on here. Okay, so I just wanted to go on to the survey, and why would we do a survey? So the main reason was that I spoke to a group. I don't know whether any of you have had any involvement with. So there used to be a group called MIST, which was Macular Impairment Support and Togetherness. I think at the moment they're not going on at the moment. And I went to talk to them and they asked me about this condition. And I have to say, I knew absolutely nothing about Charles Bonnet syndrome at the time. And this kind of, I suppose, worried me, the fact that I'd worked in vision research at this stage for nearly 20 years, and I'd actually never heard of something that it seemed to be a lot of people were affected by. So I decided last summer, I had some time, and I had a very excellent student called Claire working with me. And together we developed a survey to learn, with Fighting Blindness, I should say, as well, and with many actually participants who I really have to thank a great deal about Charles Bonnet syndrome. And what we wanted to do is we wanted to find out how many people have Charles Bonnet syndrome in the visually impaired population. But we also wanted to find out, what is it like for you to have Charles Bonnet syndrome? And at every kind of time in this, Claire spoke to focus groups, and we got, the Living with Sight Loss group to examine the survey. It's accessible and you should be able to take it as well. So I just want to describe to you maybe some of the take-homes that we have from the survey. So it seems to affect people across all ages. So people have taken it from age 18 up to age 90, and it affects people across all ages. It seems to affect males and females. However, we did have more females responding to the survey. I don't know why. That might be maybe more women have Charles-Bonnet syndrome, or perhaps it's just women like taking surveys more. I don't know. It seems to occur across all different types of eye conditions. And many of the people were registered with I should say, the Council for the Blind, the National Council for the Blind. Now, specifically what we were looking at here was that we were trying to find out about, people living with this in Ireland. And the reason for that is that Fighting Blindness is obviously an Irish charity, and we have the potential to advocate, to the government and things like that, specifically in an Irish context as well. So just kind of thinking about some more results in the survey. So over half the people who took the survey had actually experienced Charles-Bonnet syndrome. And this is much higher than any of the, well, not much higher, it's higher, a little higher than many of the surveys that are published. So the published surveys kind of said from 0.3% up to about 40%. So this is a little higher as well. And in terms of kind of what is the experiences like for the patient, so the experience really varied. So the types of things that people saw varied, the whether they were in color or black and white varied, and the frequency varied as well. So some people, these visions are continuous, versus some people, maybe they only see them once or twice a month. Now, one of the key things that we learned was actually these visions can be quite disruptive. So that's important because obviously, nobody wants a vision to come and disrupt your daily life. And that evoked a number of different feelings among patients. Sorry, here I just have some emojis here showing the feelings as well. Now, the feelings, some people were kind of amused by the visions. Other people were concerned by the visions, maybe a little bit afraid of them. So I think it probably just depends on the type maybe of vision that you have. And also what we found out was it was impacted by stress, by people feeling tired, and by deteriorating vision. So just if you were a bit stressed, if you were tired, and also if your vision had got worse, that these visual hallucinations were worse. Now, one of the main things, I guess, that we have discovered from the survey to date is just about awareness of Charles-Bonnet syndrome. So 44% of the people who took the survey hadn't actually heard about Charles-Bonnet before the survey. And it kind of seems to be maybe a hidden condition, because even out of the numbers of people who took the survey, very few of them had mentioned it to their ophthalmologists or to their GPs or any other healthcare conditions. Now, one of the positive things is that some of the people had heard about it from Fighting Blindness and from some of the support networks that they had developed through Fighting Blindness. So I think that that is very important. But we do think here that there's a key, I suppose, a difference that we can perhaps make by raising awareness. And we think that raising awareness should be, I suppose, on a number of different levels. So, for example, David Keegan and I have spoken about this, and perhaps we could have the eye clinic liaison officer talk about it to everybody who is diagnosed with a condition. And also, we would like actually to go out and talk to GPs about it a lot more, because we kind of feel that this should be something that's on the general medical education and that all GPs should know about it. So perhaps they could ask you about it or something like that when you went to it. So just, I suppose, some take homes from the survey. So number one is that we're looking for more people to take the survey, because at the moment, I think we have had 46 people take the survey, which is obviously great, but we would like if as many of you could take the survey as possible. And I've got a link here as well. Now, the survey is done via SurveyMonkey. We've had it checked for accessibility. And if you were concerned about doing the survey, it's possible that you could ring the research department and they could maybe give you a call at some stage and you could take the survey over the phone. I've also just given some links here just about, there's a little bit more information about Charles-Bonnet syndrome on the Fighting Blindness website. And there's also a wonderful charity in the UK called Esme's Umbrella, who've been doing a lot of work about Charles-Bonnet syndrome. So if you want to find out more, you could look at their website. That's Esme's Umbrella. Now, if you're ever concerned about, Charles-Bonnet syndrome or anything like that, you can always contact Fighting Blindness and ask to talk to the Insight Counselling Service. And I've given just their email address. It's insight@fightingblindness.ie, or you could call Fighting Blindness as well. And I suppose in terms of it, the take home has said we'd like you to do the survey, But we also feel, even just from the information we've got from the survey, we can maybe advocate for patients with sight loss a little bit more in terms of trying to raise awareness of Charles Bonnet syndrome. And that we can learn more, you can kind of bring this from a hidden disorder that nobody ever discussed, and they kind of, you know, maybe tolerated alone, to something that people can discuss, and, you know, it's out there in the open. And finally, I just wanted to thank a lot of people. So I just wanted to thank the research department, and particularly Laura Brady in Fighting Blindness. I wanted to thank John Delaney, who really helped us to kick off this survey, the Living with Sight Loss group, who helped us kind of beta test the survey and also advised us on kind of questions and things like that. And also I spoke recently with I think one of your connections, Coffee Mornings. And finally, actually, I want to thank the medics as well. So David and Julie and also Paul Kenna in the Eye and Ear have also been helpful in talking to us and kind of helping us to learn a little bit more about this. And then just to say thank you very much to you, because like I said, I feel like I'm talking to, I'm the non-expert here in the room, and the rest of you probably know a lot more about this and have experienced it more than myself. So thank you very much.
Thanks very much, Alison. That was great. Yeah, as Alison said, that if anyone wants to contact the research department to find out a bit more information about the survey, or to contact us on research@fightingblindness.ie, or to give the main office a call, and we can get back in touch with you. So I think we're just about right on time. So before I hand back over to Kevin to close the session today, I'd just like to thank again our four fantastic speakers, Jackie, David, Ben, and Alison. And we really appreciate you giving the time today to speak to us. Kevin, I think I'll pass over to you. Thanks very much.
Thanks very much, Niamh, and thanks to yourself and Rachel for your work on getting this webinar series up and running. And on behalf of Fighting Blindness, I think I really want to thank the presenters. They gave excellent presentations. And what I found in listening to all the presentations, that there was lots of ambition, but lots of ambition that will be realizable. And I think that's the message to take away from today's webinar, that we are an ambitious charity about making a difference to people living with sight loss. But not only are we ambitious, but through our partnerships and through the input of the clinicians and through the global partnerships with groups like Foundation Fighting Blindness and also the research institutes here in Ireland, we are making that difference. Really thank everybody for participating, and rest assured, anyone that has logged questions, we will follow up after the webinar series in relation to those and hopefully assist anyone that we can possibly assist in addressing any issues that they have. Finally, just again to thank our sponsors, Novartis, ProQR and Roche, and to hope that you all enjoy the rest of the evening. And I doubt if anything on Netflix or anything else will match the presentations this evening. So take care, and it's been really good speaking with you.